you all are attending now this session on specifically FDA expectations. Okay, and we will be discussing on the nitrosamine impurities. So hope you all attended this training session. And if I am audible to all of you, if I am visible to you, and if the slide is also visible to you, please confirm in the chat box that yes, everything is okay, so that we can continue our discussion on FD expectation on controlling nitrosamine impurities in human drugs. So please understand this topic is very important. So I just expect that anyone can confirm. So by the meantime, we will start with our routine discussion about the training session. So by the meantime, you please confirm about whether I am audible to all of you and everything is okay, and I am visible to all of you. Please confirm so that after that only I will able to start the discussion. So as you are putting the comment in the chat box, by the meantime, let us start with our the discussion on the this topic. Okay, so what we are going to discuss today on this topic. So basically, this is the overview of what we are going to discuss today. So we will discuss about the importance of topic and agenda. So very important thing is why we need to discuss these nitrosamine impurities, because now FDA is focusing so much of putting pressure, even the compliance perspective on the nitrosamine impurities. The reason is it is considered as a carcinogen. So as this product or this nitrosamine impurity is generated during the different stages of manufacturing, we need to control in the final product. So that is the reason this topic is very important with respect to the regulatory as well as compliance point of view. Even you may be aware that the products which were having the higher level of nitrosamine impurities were recalled by the FDA. Hope you are aware about all the things. So let us start our discussion on nitrosamine impurities. So many people requested me to have a training session on nitrosamine, so I am here to discuss today on this topic. So what we are going to discuss? So there will be couple of slides about the trainer, know your trainer, because the many companies consider this our training session as an external training, and they record in the training record that they attended this external training through the YouTube training program. So for that purpose, they need a CV of the trainer. So that is the reason I put the CV of the trainer, means my CV in the these slides only. So after that, we will discuss about the historical chronology and key findings by FDA. So what is the chronology? Why we are discussing about the this topic and all that we will discuss. Followed by the nitrosamine impurities understanding. So what is exact meaning of nitrosamine impurities and what type of impurities considered by the FDA, and that brief understanding we will be discussing here. Then the next point is why we are so much of concern about these impurities. There are many impurities in the drugs and drug product, but why we are so much of concerning about these impurities, that is the nitrosamine impurities, that we will be discussing with the guideline references, of course. Then what are the general root causes which can lead to the generation of a nitrosamine impurities, that we will be discussing, that what are the general root causes are there which due to which the nitrosamine impurities will be generated in the drug products. So basically that point also we will be discussing during this session. Then once we discuss about the general root causes with respect to the drugs, and if you are a manufacturer of a drug product, then what are the other sources of nitrosamine impurities other than the API, that also we are going to discuss here. Please remember this point. Many people, what they do, they only focus on the API perspectives with respect to the nitrosamine impurities compliance. So this is not the case. You need to evaluate, if you are manufacturer of a drug product or formulations, then you need to evaluate the other sources like other excipients, water and other all sources from which the potentially nitrosamine impurities may contaminate your drug product. Of course, you have to consider the API source also, but other than API also, you need to put a focus. That is the reason I considered this point during the discussion. Then FDA has suggested some acceptable intake limit, so that also I will be discussing during this session. Then there are some recommendations by FDA and how we can control these impurities in the drugs and drug product, that we will be discussing, followed by the question answer session. So if you have any questions during our training session, I request you to put your questions in the chat box. I will respond to all our questions at the end of the session. Second thing, if you need any this training presentation copy, you please put your comment in the chat box that you need a training presentation copy. I will share the link to download this training presentation copy. Okay. So today is 31st of August 2024. So today we are conducting this training session. So after this training session, if you uh, request me for this presentation copy, then I may not able to share this training presentation copy. So please, if you are interested, please put your comment in the comment section about the requirement of a training presentation copy. Then after that, we will have sharing the contact details and vote of thanks about this training session. One more point I want to highlight here, that while discussion on this topic, you know, many important point is when you get the training about this nitrosamine impurities. So what happened, most of the people, they only get confused in the different reactions, different, the structures and all. I prepared this training session in a very simple and easy to understand language. So I try to avoid all the different reactions. I try to avoid all the different structures or chemical formulas and all. I made it simple so that you will able to focus on compliance perspective. Basically, you will focus on the compliance perspective. That is what uh, this training, this is the intention of my training today. Okay. So without wasting time, let us move to the next, that is a know your trainer. So my name is Hitendra Kumar Shah. I am a CGMP compliance consultant, trainer and auditor from NADH plus GXP compliance services. I am having more than 23 or 24 years of experience in pharma and having the expertise in all the regulatory expectation like US FDA, UGMP, MHRA, TGA, TPD, NVISA, WHO, Schedule M, you know. So this is my brief profile. You can go through this. If you are interested, you can pause the video and you can go through this. Uh, till date I face US FDA 9 times and UGMP 7 times, MHRA maybe some 2-3 times, and other regulatory agency also I face the inspections. And to be very frank and to be proud that we are supporting for many companies for remediations and support for UGMP, USFDA and other regulatory agency compliance. And till date we have succeed all the customers, or you can say we have succeed all the inspections. There is not a single inspection where we face it and we and the company got the critical observations. So this is the to be very frank, this achievement I just want to share with you. So we face a lot of inspections and not a single inspection where the company received the serious observations. That is what the point I just want to share. Second thing, we are providing the third party audits also. So I have audited personally more than 500 sites till date. Even the last year we had more than 100 audits in last year. You can go through this in detail about the know your trainer. This is my major work experience. I started my career with Sun Pharma. Then I work in Blue Cross Laboratories. I work in Alembic Pharma in a 100% export oriented unit. Then I was a quality manager with the US Vitamins. Then I was a compliance head with Zydus Cadila. I was in a multinationals also where I work in AstraZeneca, Sanofi, and Mylan. And since last 6 years, I am in an independent role, in an independent compliance role, supporting the many pharma companies to upgrade, to grow, and to comply the requirements and to comply the regulatory expectations. So our company NADH GXP Compliance Services is a registered organization under the uh, small and medium enterprises under the unique number MH270137913. This is just for your information. Second thing, uh, I am Hitendra Kumar Shah. I am the approved trainer by the Government of India, and Government of India has provided this unique trainer number TP231534 to me as a trainer. This is just sharing you the information. Okay. And before moving ahead, I just want to put some disclaimer, because what happened, this is a training session. It will be recorded in the in the recorded form. So what happened, it will be available for long period in the social media. So my request is, you refer this training session as a learning purpose only. You, if you want to implement, you refer the current guideline. Okay, because we don't know, after couple of years the guideline may update, may the limits may update, expectation may be increased, so we don't know. So at this moment, whatever the current expectations are there, that I am sharing. So that is the reason, please consider this training session as a learning purpose only. Okay, you can go through the disclaimer in detail. And now, without wasting time, let us start our today's training session on the nitrosamine impurities, specifically how we can control the nitrosamine impurities in line with the FDA expectation. So when I am using the terminology as a FDA, please consider this as a US FDA. Please remember this. Okay, so whenever I'm using the word FDA, that means, unless and until I am informing, specifying anything else, it is the US FDA. Okay. So let us start our today's training session. Okay. So this is from where, we will actually discussing on the today's training session, and I request you, please carefully learn all the things, because I prepared this specifically for compliance perspective and in easy to understand language. I collected the information from so many participants, and what are their expectations, and based on their expectations I have prepared this training session. Okay. So there is some historical chronology of this nitrosamine. So what happened when, in since 2018, 2018, FDA was investigating that the presence of nitrosamine impurities in some drugs and drug product, and some drug products like ARBs, like angiotensin receptor B, or ranitidine, or nizatidine, or metformin. In that products, some, the nitrosamine impurities were identified, and they were found in an unacceptable level of the nitrosamine impurities. So this was created the start of the thought process by FDA that yes, that there are the unacceptable levels of nitrosamine impurities in such type of molecules. Then in January 2018, FDA informed the presence of impurity identified as N-nitrosodimethylamine, that is NDMA, in the ARB valsartan. ARB means angiotensin receptor blocker valsartan. Okay, hope you are aware about the ARB. Then in September 2019, FDA come to know that some common, the product which are used for a heartburn or antacid, like ranitidine, the brand is Zantac, nizatidine, which the brand is Axid, are containing unacceptable level of NDMA. So they are having a very high level of NDMA, and due to that, you may be aware that so many products were recalled which contain the ranitidine, or nizatidine, or even the valsartan, and so many products were recalled, right? After that, what happened in December 2019, FDA come to know that the metformin is a normal common drug, right, for treating the, or for a treatment of the diabetes, right? So metformin is a diabetes medicine, and it was also observed that some, it was suspected that some NDMA impurities are there in the metformin product. That is what the FDA got the idea. But when FDA tested the metformin product, metformin, the immediate release product, in February 2020, and they come to know that the NDMA impurities are there, but they are within the limit. Okay, within the acceptable limit. So they don't took it more seriously. But then in the month of May 2020, when the extended release metformin formulations were tested, and they come to know that the NDMA impurities are higher than the acceptable intake limit, and then even the metformin product was also recalled from the market. So due to this, you may be aware, if you are in a pharma company, if you are working in the pharma, you may be aware that there were a lot of recalls of so many drug products as well as the drugs in the year 2020 to 2022 due to this nitrosamine impurities. Now most of the companies changed their process, developed their process, modified their processes, and they took so much of action to control the nitrosamine impurities and to monitor the nitrosamine impurities in their drugs and drug product, right? So this is what the historical chronology of the nitrosamine impurities. Okay, again I'm telling you, whatever I am discussing today, it is based on the FDA recommendation. Okay, this is not my thoughts, please remember this. This is not my thought. Whatever I am sharing you, all the information is in line with the US FDA expectations. Please remember this word. These are not my personal thought or personal opinion. I am very clear with this. Okay, if I am speaking very fast, I suggest you can reduce the speed or you can go back and again you can learn the same thing. You don't skip the any content. Many people, what they do, they just go on reading the content present on the slides, and they consider that they understood. But the point is, in the slide I am putting some content, but other than that I am discussing more and more points. Okay, so during this all evaluation and all, there are some key findings by the FDA. What are the key findings? So FDA evaluated that the processes that use common amines in API synthesis, and also learned that the common synthetic pathways could also introduce other types of nitrosamine impurities besides NDMA. So FDA come to know that when you are using the common amines, there is a possibility of generation of a NDMA. That is the first key finding. Second thing, if you are following some synthetic routes, that some synthetic routes, some reactions are common for different API manufacturing. So if you are having these common synthetic routes, then definitely there may be possibility of a NDMA impurity generation. Third point is that there is not only one nitrosamine impurity, that is NDMA, but there are the other nitrosamine impurities also in addition to the NDMA. So that also FDA identified from this their studies. Okay, then second point, what FDA come to know that ranitidine products stored at room temperature, even at initially they may have the impurities within the limit or within the specification or acceptable intake limit, but on prolonged period when they are stored at a room temperature, there will be increase in the impurity level. Okay, so FDA preliminary results using accelerated stability testing demonstrated that elevated levels of NDMA were measured in all products after 2 weeks at accelerated condition. After 2 weeks, okay, at a accelerated condition means at a 40 degrees centigrade and 75% RH. 40 degrees centigrade temperature and 75 degrees RH, when the product was stored for about 2 weeks, then the NDMA impurities were found to be increased. So these are the key findings by the FDA. So what does it mean? That is now I am sharing you. That is what I'm telling you. Just don't read all the things. Of course you read all the things, but also you listen what I'm explaining to you. That is what the point I want to explain you clearly. So what happened when these expectations come into picture and the company started changing the routes of synthesis and changing the lot of processes in the manufacturing processes and all. So the company started claiming that they are complying to the NDMA requirement. It is within the limit, or NDMA is not there in the product. Then the FDA suggested that check the accelerated condition data, because at the accelerated condition also these impurities are found to be likely to increase. That is what the point I want to clarify here. So the point here is, when you are manufacturing this type of products, right, like metformin or ARB, valsartans and all, ranitidine and all, Then the NDMA impurity testing, if it is possible in the product process based on the pathway and all, then you have to consider this as a stability indicating method to prove that there is no increase in the limit on the prolonged long-term stability study conditions. Hope you got my point here. Many companies are not considering this and they will likely to get the problem or non-conformity in the future. Please remember this, okay? I am very clear the expectations by the FDA and I am making it very simple language so that you will understand in a better way. So now let us understand what is actually nitrosamine impurities. People are telling nitrosamine impurity, nitrosamine impurity. So what exactly is the meaning of a nitrosamine impurity? So basically the terminology nitrosamine, it is a class of the compound with the chemical structure of a nitroso group which is bonded with the amine group. So the structure is given here in the picture, also the structure is there. So you will come to know that a nitroso group and it is bonded to the amine group. So it is called a nitrosamine. Okay. The compound can form by nitrosating reaction between amines and nitrous acid. So if in your process, manufacturing process, if you are the manufacturer of the API and if you are having the manufacturing process where there is a reaction of the compounds of amines with the nitrous acid, then there is a possibility of a generation of a nitrosamine. You got my point? Because under the acidic conditions, these impurities are increased. I am making in a very easy language. Okay, I am not. I am not here to just explain and confuse you. I want to clear this thought. That is what my intention on today's training session. So FD identified that uh, we are discussing about NDMA impurity and nitrosodimethylamine, right? But there are the other nitrosamine impurities also. Which are these? NDEA, NMBA, NIPEA, NDIPA, NDBA, NMPA. So these are some additional 6 nitrosamine impurities are there which are generated as a part of the different reaction and which are potential carcinogen. So our product should be protected from these impurities. That is what the point FDA is clarifying to all of you, and you have to ensure that your product should be free from all these impurities. So now point is, what is the problem? What is the concern with these impurities? Earlier, we were not bothered about these impurities, nitrosamine impurities, and suddenly today we are bothering about these impurities. So what is the problem? So basically what happened, the nitrosamine compounds are the potent genotoxin agent in uh, animal species. So specifically they are studied on the different rodents and they found that they are possible human carcinogen, and it is proved by the International Agency for Research on Cancer, IARC institute. So that institute confirmed, this agency confirmed that these nitrosamine compounds are the potential carcinogen in many other species and they are potential carcinogen for human also, and that is the reason there is a more concern. These basically these molecules are referred as a cohort of concern. What happened? No, as per MS guideline M7R1, assessment and control of DNA reactive impurities in the pharmaceuticals to limit potential carcinogenal risk. This is the guideline M7R1. Under that, it is considered as a cohort of concern. Means what? These molecules act on the our chromosomes. Our chromosomes are made of a DNA, deoxyribose nucleic acid. So if you check the DNA structure, it contains the uh, different nitrogen bonding. Okay, adenine, thymine, guanine, cytosines. All this contains the nitrogen bondings, and these nitrosamine impurities, which are also having this, these nitrosamine impurities act on these bondings and can lead to the change in the DNA and ultimately can lead to the cancer. You got my point? This is the thing, and that is the reason that International Agency for Research on Cancer claim this as a potential carcinogen for the human, and that is the reason now all the pharma regulatory agencies, not only the US FDA, all the TPD, means Canada agency, and all the other agencies are focusing now on this nitrosamine impurities to control in the drugs and drug product to safeguard the human. Okay, so hope you understood the brief about the things, right? Now the point is, what are the causes now? Okay, what are the causes or root causes which can lead to the generation of these impurities? What are the causes? What are the root causes? So there are some causes are suggested by FDA. So we will discuss about these causes, and after that we will discuss about how we can remediate and what are the expectations of the FDA during this session. So there are some general conditions that lead to nitrosamine formation. So there are some general conditions are there. What are the general conditions? So if you are using the secondary, tertiary or quaternary amines and nitrite salts under the acidic reaction condition. So just now I discussed about the amines with the nitrite salts under an acidic condition. So it can lead to the generation of the nitrosamine. Just now we discussed, right? Second is nitrites used as reagents in one step, in any step, which can carry over into the subsequent step. So if you are using the nitrites as a reagent and it is carry over in the next reagent, and during purification even you cannot remove it, then it can react with amines and can generate the nitrosamine. So if you are adding the nitrites, then for particular reaction, then immediately you need to have immediately to control the nitrites, so that the nitrites can be removed from the next remaining or unreacted nitrites from the next reaction, before the next reaction, so that the remaining nitrite will not convert into the nitrosamine impurities. So you have to remove it immediately. Okay, so if you are not removing also, then it can lead to the nitrosamine impurities, right? Hope I am loud and clear and everybody can able to hear my voice. They can see me and everything is clear. Slides, they can see the slides also. Okay. The second root cause, what FDA is suggesting, that the sources of amines what we are using. So what are the sources for amines like secondary, tertiary and quaternary amines which can form the nitrosamine? So basically when the API or the degradants or intermediates or the starting materials which are used for manufacturing of the API, if they contain the secondary or tertiary amine functional group, then also there may be possibility of generation of a nitrosamine. Because what happen, if you are manufacturing the API, you may be using some intermediates or you may be using some starting materials and all. So in that materials, if there is this amine group, then there is a possibility, right? Or there may be possibility that some API degradants also can lead to the nitrosamine. So these all points you have to consider. Okay, and please remember these root causes, because after understanding these root causes, at the end of this session we will be focusing on how we can control it. So that is why it is very, very important. You got my point? Second is, tertiary and quaternary amines may also be added intentionally as a reagents or catalyst. Sometimes during the reactions, if it is required, then tertiary or quaternary amines are added as the reagents or the catalyst, and it can also there may be possibility of a generation of these impurities. Amide solvents. So if you are using the amide solvents which are susceptible to degradation under some conditions, then in that case also it will be the source for the secondary amines. So what happened when the amide solvents are degraded under some reactions, then it will convert into the secondary amines, and after secondary amines conversion, after reaction with the further groups, it can lead to the these impurities. Secondary amines could also be present as impurities in the amide solvent, or in some cases if you are using the amide solvents and if you are testing the amide solvents, you have to check whether there are some secondary amines as impurities or not. So because amide solvents may not getting degraded, but the amide solvents itself will contain some low concentration of secondary amines, which can be converted into further the nitrosamine. So these are all potential, you can say, possibility of the root causes. Okay, so my point is, remember this all root causes, all record it, so that based on all the root causes you have to evaluate whichever is suitable for your organization level, and based on that you can put the control plans in place to control the limit or the level of the impurities in your product. Okay, the next is, next is contamination in the vendor sourced raw material. Very important. So if you are using, see, all materials cannot be manufactured by the one company, right? So if you are manufacturer of the API, you need to source so much of so many solvents, reagents and all from the vendor. So if you are sourcing some materials, nitrosamine impurities can be introduced when you are sourcing some starting materials, raw materials are contaminated with these impurities. You got my point? So if your product is susceptible for these impurities, you have to evaluate your all input materials also. Very clear message, huh? And again I'm telling you, this I am not telling this to you, this is expectation by the US FDA. Again I am sharing from the bottom of my heart that please remember this. Many people, what they do, they do the risk assessment of the process and they prove that there is no possibility of a nitrosamine impurities in their product. And how they do the risk assessment? They copy the risk assessment from one company and they prepare in their format. Absolutely wrong practices. Many people put in the WhatsApp groups. There are so many WhatsApp groups or in the social media and all, that do you have the nitrosamine risk assessment? Please share the nitrosamine risk assessment. And they take the nitrosamine risk assessment and prepare same to same for their plant. They don't know how the process, what are the reagents, what are the potential sources in their product and all. So please remember, my dear friends, please remember this point. Okay, nitrosamine contamination has occurred when fresh solvents like orthoxylene, toluene, methyl chloride, these are contaminated during the shipment from the vendors. So what happened? Many vendors, what happened? They ship these solvents and they call it as a fresh solvent. Okay, these are not a recovered solvent. I am not discussing about the recovered solvent at this moment. So they claim that these are the fresh solvent and they fill the, what the companies are doing, they take the these solvents in a container or the big storage vessels, and from the big storage vessels they transfer into the drums and they dispatch the drums to the different drums or containers, and they dispatch these 250 liters containers to the different companies. Now in that case, due to these transfers and the shipments and all, there may be possibility of a contamination of these fresh solvents. These are the fresh solvents, but during transfers and all, there may be possibility of a contamination and in the solvent. So you have to evaluate your solvents for nitrosamine impurities. So I have evaluated most of the more than 500 sites till date. I have reviewed the nitrosamine impurity evaluation done by the many companies. Surprisingly, these solvents are not evaluated by many companies. They just specify that these solvents are the fresh solvents. We are not using recovered solvents. My dear friends, if you are using the fresh solvents, at least you have to evaluate initial shipments and all, that whether they are contaminated with the impurities or not. But most of the companies not evaluated. If you have evaluated, then very good. But if you have not evaluated, please start evaluating. At least you evaluate 3 lots and you at least ensure that the solvents are not getting contaminated. Okay, as a part of the vendor evaluation process also you can evaluate these. I don't have any problem. As a vendor evaluation also you evaluate. If you have not evaluated as a part of the risk management, no problem, but you can evaluate as a part of vendor evaluation also, no problem. Then the next is what, with respect to the contamination, is a sodium nitrite. So what happened? Sodium nitrite is example is given by even FDA only. This is example by FDA. So sodium nitrite is a known impurity in some starting materials like sodium azide and all, and it may be present and react with amines under acidic conditions to form a nitrosamine. Similarly, in sodium nitrite, potassium nitrite also can lead to the generation of impurities. So if you are using the sodium nitrite or the potassium nitrites, then be cautious and evaluate in detail about the nitrosamine impurity level in your product. Secondary or tertiary amines have been reported as impurities in some raw materials. Just now we discussed, but as is a part of contamination, I consider this point because this impurity also may be contaminated by the secondary and tertiary amines. So I thought as a part of cross contamination and contamination, we discuss this point here. Hope I am not so fast. Okay, if I am fast, you please put comment. I will try to speak slow so that everyone should understand easily. Otherwise you can reduce the speed. In the YouTube you have the provision to reduce the speed 0.75, 0.75 you can make the speed, so that you will get the slow speed. Okay, because what happened, I need to cover this whole session in 1 hour or 1 hour 10 minutes, so that is the reason I am speaking in a fast. Okay, starting materials which are outsourced or intermediates may be at risk through cross contamination if they are manufactured at sites where nitrosamine impurities are produced in other processes. Yeah, this is also the one point, that most of the companies, they purchase the starting material or intermediates from the other vendors. So other vendors, what happen, the product is safe. Okay, product, there is no possibility of nitrosamine impurities and all, but the equipments which are used, that equipment may be used for the other products, where in that product all these amines, no, secondary, tertiary or quaternary amines may be used, or some reactions will be done which can lead to the generation of the nitrosamine impurities, and from that these intermediates or the starting materials may be contaminated with these impurities. So if you are the API manufacturer and your manufacturing process may be proving that there is no possibility of generation of nitrosamine impurities, or you are not using any amines and all in the reaction, but still you need to evaluate the starting materials and all. There may be possibility of contamination of your starting materials and intermediates with the nitrosamine impurities, right? Hope you are with me, and again I am requesting, if you have any questions, please put your questions in the chat box. At the end of the session I will respond to your all queries. If you are interested in the training presentation also, you please put your comment. I will share the training presentation to all of you. Okay, the next point with respect to the root cause due to which nitrosamine impurities will be generated, and that is the cause is a recovered solvents, catalyst or reagents. So if you are using any recovered solvent, catalyst or reagents, then you need to think that there may be possibility, and that possibility you need to evaluate. Okay, that there may be possibility of generation of impurities. Okay, so what are the possibilities? Presence of residual amines like triethylamines or diisopropylethylamine. So there may be presence of residual amines in the solvents or residual recovered solvents. If the recovery process involves quenching step, then also the nitrosamine impurities may be generated. So in quenching step, what happened, the nitrous acid is used to decompose or degrade the residual azide, and it is cooled immediately, and due to that also there may be possibility of a generation of impurities. And the second point is what happened, there may be possibility that if you are recovering the solvent at specific temperature, the boiling point of these impurities, NDMA and NDA impurity, is 151 to 153 degrees and 175 to 177 degrees. Means what? NDMA impurity boiling point is 151 to 153, and NDEA impurity boiling point is 175 to 177 degree. So what happens if you are recovering the solvent and you are recovering the solvent at particular temperature, then along with the solvent, these impurities will also be recovered and added in the recovered solvent. You got my point? Because the process doesn't identify whether you are recovering solvents or impurity. Process only recover based on the boiling point of a particular solvent. So it is boiled and then it is condensed. So along with the solvent, this impurity will also be converted in the solvent form, and automatically it will be condensed in the form of a liquid form in the specific recovered solvent. So you have to consider this point also when you are using the recovered solvent in your organization. Now why I am discussing this all point, when you will perform the risk management, you have to consider all these points. I have seen many companies that these all points are not considered during the risk management of a risk management in your for your API for the drugs or API for the nitrosamine impurities. Okay, so all points you need to cover, you need to consider. Then other point is what? API manufactured by two different routes. This is very important. Correct, correct. See what happened if you are manufacturing two different API, sorry, one API but two different routes. Route of synthesis, A route of synthesis and B route of synthesis. Now you know that route of synthesis, due to the route of synthesis A, there is a generation of some nitrosamine impurities. Okay, so you are using the route B, where route B is not generating the nitrosamine impurities. Now what happened? Most of the company, they have the procedure to use the solvent for the same product. So if you are manufacturing the product and for recovery or some process, if you are using the you want to use the reuse the solvent, so the company claims that they are reusing the same solvent which is used for the same product. That is what the companies are telling. But if the solvent is used in the route of synthesis A, where the nitrosamine impurity is generated, that solvent if you are using in the route of synthesis B, then definitely there will be the contamination in the route of synthesis B, and your product will be contaminated with the nitrosamine impurities. So now you will be surprised that why your product is containing nitrosamine impurities, because your process is a very good, your process is not generating the nitrosamine impurities, but you are using the recovered solvent or used solvent which is generated in the stage A. That you need to remember. Recovery by the third party contractors, yeah, this is again the big problem. Most of the company, what happened, they don't have the solvent recovery plant. So what happened, they give all the solvents to the third party or third party contractors, and they recover the solvent and they will again return back to you. Now here the problem is, what is the control on these third party contractors? They may not be recovering the solvents received by your company only. They may be getting the solvents from multiple companies. So if any company solvent containing the nitrosamine impurity or any other impurities, there are 7 impurities are there, right? Not only 1 or 2 or 3. There are 7 impurities, my dear friends. So any impurity is there, ultimately it will contaminate your solvent also. So you have to put a lot of controls if you are using the third party contractors for using the recovery for recovering the solvents. The next is lack of validated equipment cleaning procedures. Now the here point is, most of the equipments, if you don't have the cleaning validation, means validated cleaning procedure, then it can lead to generation of impurities. Impurities, wherever I am discussing today impurities, it is a nitrosamine impurities only. Okay. So example is given like orthoxylene and toluene were found contaminated during the recovery due to the inadequately cleaning. So if you go on recovering these solvents continuous basis, orthoxylene and toluene, and if there is a orthoxylene and toluene, so the changeovers are not clean properly. So when these solvents come together, then there is a possibility of generation of nitrosamine impurities, because there is no validated cleaning between the product changeover. Hope you are clear with these things. Okay. I suggest, there will be some 15 minutes more, miss 1 hour 15 or 20 minutes required for this training session. So please be with me. This topic is big, so I don't want to skip this topic, and I don't want to keep it small. Okay. So that is the reason I am just requesting, please be with me. The next is quenching process as a source of nitrosamine contamination. So basically, nitrosamine is generated due to the quenching step, where nitrous acid is added to the reaction mixture to decompose the residual azide and can lead to the nitrosamine. I am trying to finish this within the period, but please bear with me. Okay. 6 is lack of process optimization and control. So if your process operations, manufacturing operations are not optimized and not controlled, then also there may be possibility of a nitrosamine impurities. So these are all points you have to consider during the risk management, whatever points I am enlisting here. So what are the points? Lack of optimization of the manufacturing process for API, like pH, temperature and all. Even why these points are identified, because FDA has identified that where the reaction conditions, there is a big variations, there are no optimized process between the batches, there is a possibility of generation of these impurities. This is what FDA identified, and that is what I considered here, this point. Okay, you got my point. This is not my words. FDA identified these are the issues, and that is the reason I consider this point. So this you can consider, and uh, for the performing the risk management. So basically what happened, these are some general root causes. Okay, so this list, whatever we discuss about the sources, is not exhaustive. You have to evaluate further the root causes or possible causes in your organization with respect to the process, input raw materials, controls in your vendors, and practices, and controls on your operations, and other input materials other than API, so that you will able to evaluate the additional root causes also. Okay. Right. Now this is what we discuss about the API. But if you are the drug product manufacturer, then other than API, what you have to evaluate, that is what the most important question. Most of the drug product manufacturers, what they do, they just take the declaration by the API manufacturer, and they claim that it is a nitrosamine impurity within the acceptable limit, or there is no possibility of nitrosamine impurity in the drugs. That's it. But what about the excipients? Because nitrites are common nitrosetting impurities that have been reported in many excipients at ppm levels. This information is clearly specified by FDA. So if you are using the excipients for your drug products, evaluate your excipients also. Second is, nitrite impurities are found in the range of commonly used excipients. So whatever the common used excipients are there, which are manufactured by synthetic origin, you have to evaluate clearly. Then you have to strictly follow the supplier qualification program, where just like how you evaluate, or you may have some questionnaire with respect to the nitrosamine impurity evaluation for API. Similar type of questionnaires you have to you should use for evaluation of your excipients also. Many companies, even they don't focus on the excipients. But remember, one very important point is, if your drug product, let us say the tablet formulation, the dose or the tablet unit weight may be about, suppose, 500 milligram, in that the API may be hardly 50 to 100 milligram and more than API, the excipients will be in the drug product. And if the excipients contain your nitrosamine impurities, then your drug product is contaminated with these impurities. So many companies only focus on vendor evaluation for API. They don't focus on evaluation for excipients. So this point you should consider while evaluating the excipients also or excipient supplier also. Drug product manufacturers should also be aware that these impurities may be present in potable water. So if you are using the potable water from the open source like seashore or the underground bore water, that potable water also you need to evaluate whether these impurities are there in the water. Because what happened, whatever the purified water is used for manufacturing of the product, then that purified water will remove the different impurities, but they may not remove the nitrosamine impurities. Second thing, if you are using the ion exchange, okay, cation exchanger, anion exchanger and all, so this also can lead to the increase in the nitrosamine impurities in the water. So you have to evaluate your potable water also. But many companies, I found that they are not evaluating this water, and due to that there may be possibility of increase in nitrosamine impurities in their products. Some products may undergo degradation pathways that form nitrosamine impurities. This could potentially occur during the drug product storage. So just now I gave the example that FDA proved that the ranitidine product on storage on a long-term period, it lead to the increase in nitrosamine impurity content. So that you need to consider for the drug products, whether they are you are storing for long period, whether during the storage, if there is an increase in nitrosamine impurity during the storage, that you have to evaluate. There are some FDA recommendations that FDA recommend that manufacturers consider the potential causes of a nitrosamine impurity formation, and then you have to take the actions. So you have to identify the what are the possibility of a nitrosamine impurity generation, that you have to identify the risk, and then you have to take the action. So whatever we discuss up to here, you have to consider all these points. And addition to that, you have to consider the additional point which may be suitable for your process equipments and other than equipments like the other input materials and all, and then you have to evaluate it in detail, right? And then you, on the basis of that, you have to take action. Manufacturers should prioritize evaluation of APIs, drug product based on factors such as maximum daily dose, duration of a treatment, therapeutic indications, and number of patients treated. So what happened, based on the if you are the drug product manufacturer, then based on if you are manufacturer of the APIs and drug products, then you have to evaluate what is the duration of a treatment and what is the maximum maximum dose, so that based on that you can prioritize the actions. For example, if it is a long duration treatment is for long duration, then your action should be on priority. Or if your maximum daily dose is higher, then also you have to take actions on the priority basis. That is the way you have to put the actions. As the new information becomes available and FDA's understanding of nitrosamine in drugs evolves, the agency may recommend that certain drug products become higher priorities for risk assessment. So at this moment, some drugs are identified and some pathways are identified. The reactions are identified which can lead to the generation of a nitrosamine impurities. But FDA is still working on that, and if any updates are there, FDA will update to us. Then the next recommendation is you have to perform the risk management as per the ICH Q9R1, and you have to identify all the potential possibilities of generating or transmitting the nitrosamine impurities in your drugs and drug product, and based on that you can take the mitigation action in the mitigation action to control the these impurities, right? These are FDA expectations, so I'm sharing you. Again, I'm telling you, whenever I'm using the word FDA, please remember that FDA means the US FDA, right? So further on the recommendation by FDA, I would say that FDA has given some recommendations about the acceptable intake in nanogram per day. So basically these impurities are there, the listed impurities, and in the table you can see this table is visible to all of you, and in the table the nitrosamine impurities are listed and there is the acceptable limit in the nanogram per day. Okay. So here the maximum potent impurities limit is 26.5 nanogram per day. That is what the point here I want to make you clear. These limits are okay if your product is containing the only single nitrosamine impurity. But if your product contains more than one nitrosamine impurities, then the total of this impurity level should not exceed 26.5 nanogram per day based on the maximum daily dose. Based on maximum daily dose, my dear friend, please remember this. Means what? Suppose your API dose is suppose 5 milligram per tablet. Okay, let us say example. So if you are manufacturing the dose 5 milligram per tablet and maximum daily dose is suppose 20 milligram per day. So if in a 5 milligram of API, if there is suppose 10 nanogram, then per day will be 40 nanograms. So it will cross the 26.5, right? So 26.5, whatever the limit is there, it is a maximum daily dose. Please remember this. This is not in a particular API or drug product. It is based on the maximum daily dose limit. Please remember this. So many people, what happened, they test the API and they prove that it is below 26.5. My dear friend, if it is below 26.5, if it is a 20 nanogram per day also, and if it is a one tablet per day, then it is okay. But if it is a two tablets per day is the dose, then it will cross the acceptable intake limit and you are not complying to the requirement. Hope you got my point. This point is very clear, so I want to make it very clear. In some companies I found that they send the sample to external laboratory, contract laboratory, they give the results that this impurity contains 5 nanogram or 10 nanograms, 8 nanograms and all, and they claim that it is within the FDA recommended limit or AI limit. But my dear friend, remember that it is based on the maximum daily dose. Hope my message is loud and clear, right? So you have to consider the maximum daily dose and then only you should put that limit, right? So if nitrosamines without published AI limits are found in drug products, manufacturers should use the approach outlined in ICH M7R1 to determine the risk associated with the nitrosamine and contact the agency about the acceptability of the proposed limit. So you have to go through this ICH M7R1 guideline, and because there are some other nitrosamine impurities also, potential nitrosamine impurities, and where the limit is not recommended by FDA, then you have to evaluate it and you have to update this to the FDA about this impurity. Second thing is there are some other molecules also. Okay, so other molecules, NDSRIs, so where there are the FDA has recommended the limit, acceptable limit for different molecules. So you can go through this link where you will find that there is a link about the total number of molecules, different molecules, and what is their impurity level for this nitrosamines. So that is given by the FDA. So you can go through this link. There are so many molecules, so I didn't put here, but you can definitely go through the link and you will get the better idea. Okay, so now the question is how we can control. That is the very important thing. Up till here we were discussing about what are the actions and all, what are the type of what is the nitrosamine impurities and what are the possibilities and all we were discussing, right? Now the point is how we can control. Correct? This is the main point, right? So there are some points FDA has suggested, and further you can, this is again I am just giving the steps, but you can consider more also. Okay, assess the risk of nitrosamine impurities in APIs, marketed products, and products under approved and pending applications. So whatever the products you submitted but waiting for approval, or which are under approval, or which are in the market, whatever is the product, all type of products, you have to evaluate the product for the nitrosamine impurities, and you have to assess the risk. Okay, manufacturers, now whenever you are doing assessment and all, FDA is very clear that this assessment is not required to submit to the agency. So you will perform the risk assessment for the product. Either it is a submitted product, or either it is a under approval product, or it is a marketed product, whatever. So the assessment is not required to submit to the agency. You can keep it with you, but if it is requested or required by the FDA will communicate to you. Then you should able to produce the document. You should able to submit a document either to the agency or during the inspection, whatever is the thing. So you should perform the assessment, but no need to share to the FDA agency. This message is very clear, so I thought I will put this message in the part of the presentation. Okay, then second is conduct the confirmatory testing when there is any risk for the presence of nitrosamine impurities. So when you evaluate your all processes, raw material vendors, and all the solvents, recovered solvents, water and other reagents, catalyst, everything, equipment cleanings and all the points, when you evaluate and you come to know that yes, there may be possibility of a generation of a nitrosamine impurities, then you have to conduct the testing of your product for nitrosamine impurities, right? Report the changes implemented to prevent or reduce the nitrosamine impurities in API and drug products to the FDA. So suppose based on the evaluation, you make some changes in your process, or make some changes in your products, or make some changes in your synthesis, or make some changes in your vendors and all, then you have to inform, or you have to report these changes to the FDA, right? Optimize the design of the manufacturing process for APIs during the route of synthesis development to minimize or prevent the formation of nitrosamine impurities. Because as we discussed that due to the lack of optimization and control also, these nitrosamine impurities can increase. So if so how you can control, you have to optimize the design of a manufacturing process for API during the route of synthesis, and minimize the nitrosamine impurity generation, right? Hope this point is very simple, and I am making it you more simple way, so that you everybody will understand in a simple language, right? Then the next is how we can control. If any root cause as discussed above in the general root causes, then take the actions. So we discussed about the general root causes, right? 1 to 6. You remember? Use of a secondary, tertiary, and quaternary amines, then the quenching process, and sources, then input raw materials, recovered solvent. We discussed a lot of possible causes, right? So that during this evaluation of these possible causes, if you identify any cause in your process, or in your plant, in your manufacturing facility, then that against that also you have to take action to control the nitrosamine impurity generation in your product. Drug product manufacturers should conduct risk assessment to determine the potential for nitrosamine impurities in drug products. So if you are manufacturer of a drug product, so you have to perform the risk assessment. So you don't only need to take the declaration by the APIs, but you have to perform the risk assessment if you are manufacturer of a drug product, right? A risk assessment should involve collaboration with API manufacturer to aid in the identification of the API route of synthesis or other process conditions of the API manufacturer that put the drug product at risk for the nitrosamine impurities. So many drug product manufacturers, they just take the declaration. So my point is they have to perform the thorough risk assessment on the nitrosamine impurity evaluation. Okay, if a risk of nitrosamines in a drug product is identified, then you have to test the batch for nitrosamine impurities, and the method you are testing method you are using for nitrosamine impurities, they should be sensitive, they should able to detect and quantify the residue within the limit, and the method should be testing method should be validated. These are some clear control mechanisms are recommended by the FDA. Again, I am telling you, these are the recommended by the FDA. You are with me and you understand now all the points. So before closing this period, I just want to share you information that if anyone interested in the certificate on this training session, on our training certificate, even the trainer number will be appear, then you should be the part of the CGMP and Advanced GMP membership program. So in the YouTube membership program, you can click the Hitendra Kumar Shaha. Once you click the Hitendra Kumar Shaha, you will find the join button. And once you hit this join button, there is a three different membership program: GMP, CGMP, and Advanced GMP. So the members who are the part of CGMP and Advanced GMP, they are entitled to get the certification. See, the GMP program, the only GMP program membership the people will have, they will not get the certification. This is the part of YouTube. You are going to pay this some small amount to YouTube only, okay, so that we can generate the training certificate for CGMP and Advanced GMP members. And of course, along with the training certificates, the memberships will have the additional benefits like discounts on the training programs, paid training programs, seminar, free doubt clearing sessions, and GMP updates and all. So based on the different kind of a membership program, GMP, CGMP, and Advanced GMP, you will have a different facilities or different benefits. So once you click this join button, there is a recorded video by me. I put it specifically for you so that you will understand what are the different benefits under GMP, what are the different benefits under CGMP, and what are different benefits under Advanced GMP. Many people are interested in a one-to-one interaction to me and understand or clarify some doubts. For them, I preferably generated this Advanced GMP membership program so they can discuss monthly basis with me personally and I can clarify their doubts. So you can access this membership program, and based on that there is a video, you can go through this. Once you click this join button, automatically after clicking this join button, you will be taken to the short video. You can go through that video to get the clear idea about this membership program. This is prepared with the help of YouTube for your sake of benefit. You got my point? And you are going to pay the fees to YouTube only. This is what it is clear. If anybody demands the payment either in a cash or online transfer, please don't pay any fees to anybody. You can click and you can pay directly to the YouTube only. There is a link in the YouTube. In the YouTube only you have to pay the fees. Very clear. Okay. This is what from my side. Hope everyone understand all the points clearly. These are my contact details. If you are interested in any our services like training, site training programs, like we conduct the on-site training program, we can come to your site and we can deliver the training sessions, or third-party audit services, or if your company is going to face any regulatory inspections, then definitely we can support you for our benefit of your organizations. Thank you so much. You can go through this website www.nad@gnp.com, or you can contact through these numbers given below. Thank you so much, and ensure all time compliance. Thank you so much. Now let us discuss on the questions, and I will clarify your all questions. So I understand that 10 minutes are there, or 10 minutes are more, but I will quickly go through this. Mr. Meladur Rahman is asking, dear sir, uh, does material other than API manufacturer is bound to share NDMA statement to finished product manufacturer? Yes, I suggest that you have to prepare the questionnaires based on the expectation, because what happened, the other than API manufacturer, um, excipient manufacturers, some excipient manufacturers, they are organic, starch or microcrystalline cellulose and all, so they are not a manufacturing on a synthetic basis. So but they may be using some reagents, so you can prepare the questionnaires that whether they are using this type of chemicals and all, and you can get it from them. But based on that you can take the statement from the uh, excipient manufacturers, that whether there is a possibility of nitrosamine impurity. Not, don't put the name NDMA, put the nitrosamine impurity statement, and based on that uh, you can evaluate your drug product. Okay. Rutuja Tilve is asking, kindly share me the copy of the presentation. Yes, Rutuja, just a minute. I will share the link, and I have shared the link. So uh, you can use this link to download the training presentation. Remember, the link will expire in couple of days, so please download ASAP. Okay, so I have shared the link, please download it immediately, right? Then uh, Utakuru is asking, Utakuru Nagaraja is asking that please share the PPT. Yes, I have shared the PPT. Please, in the form of link, please download it immediately. Mr. Manikandan is asking, sir, please make a session about the QRM for excipients. Uh, okay, okay, no problem. I will definitely prepare this session. Okay. Isha Shah is asking, hello sir, please give information about membership program. Yes, Isha, what you can do, you can click the join button. You can go through the Hitendra Kumar Shah in the YouTube. Once you go through Hitendra Kumar Shah in the YouTube, then you will find that there is a join button. You click the join button, there is a separate video where all the membership benefits are explained by me, so you can go through that and you can get the benefit of a membership program. Okay, and remember that don't pay the membership program fees to anybody, it should be paid to the YouTube only. Okay. uh, Mr. Virendra Jagtap is asking, good evening. Could you please present with one example for product with entire formulation? Yeah, definitely. But what happened? No, we have a limited time, and this training still it cover more than 13-15 minutes are over. So that is the reason I could not take the things. But wherever possible, I consider the different reactions, different reagents name, different solvents name, where there is a possibility of a nitrosamine impurities. Okay, no problem. I will consider this. No problem. Mr. Nikhil is asking training PPT. Yes, Mr. Nikhil, I have shared it. And Mr. Nikhil, I think you are the, I think part of a membership program. So if you are the CGMP membership program or advanced, you are the member of a CGMP or advanced GMP, then you send me the email or you contact me through email or WhatsApp personally. And if you are the part of a CGMP and advanced GMP only, huh? In case of a GMP program, you will not get the certification, but if you are the member of a CGMP and advanced GMP, then only you will get the certification. So you can just verify it. Here I am just able to see that you are the member. So I am just sharing a thought. Okay. So you can take the certificate. Okay. We will issue the certificate. Miladur Rahman is asking, if any raw material manufactured say that in their material all the chances of NDMA presence is low, in that case what should I do to use it in the formulation as a finished product manufacturer? My dear friend, if they are claiming that NDMA presence is low, they are not telling the NO. They are telling low. That means there is a chances of a NDMA impurity. So NDMA impurity level you need to evaluate based on your drug product maximum daily dose. So if their NDMA impurity is low, for example only 5 nanogram, but if that excipient is used such a way that consumption will be 4 times, then it will be more, or 10 times, then it will be 50 nanograms. So it will cross the AI limit. So you have to test, you have to evaluate, or you have to evaluate the vendor, or you have to control the nitrosamine impurities in the excipients. Okay. Miladur Rahman is asking, what is the quenching step? See, basically in the quenching step, when you are applying a very high temperature, and after high temperature you are suddenly reducing the temperature, means from very high temperature to very low temperature, this is called the quenching step. Okay. Dear sir, is it mandatory to perform NDMA risk assessment for finished product manufacturer for all finished product? Yes, for all finished product you have to evaluate the NDMA risk assessment. Actually, NDM, not the NDMA impurity assessment. You should use the nitrosamine impurity risk assessment. Under nitrosamine impurity, there will be multiple impurities, right? There is not only NDMA impurity. There are so many other nitrosamine impurities. So you have to perform the risk assessment for all the products. Remember this. And which are susceptible to NDMA generation, for that you have to test and you have to prove. But if there is no susceptible, there is no possibility of a nitrosamine impurity and all, how you will come to know? Based on the risk assessment for all the drug products only. Okay. Gowar Raja Vandanapu is asking, sir, are you going to keep this class in your YouTube channel? Automatically this copy, whatever I am discussing, there will be a copy in the YouTube with the same link. Whatever you join today, with the same link you will able to access this training program in the YouTube. Same link will be working. Okay. Mr. Satish Kumar asking, 3 batches are need to test. Yeah, you can see, again I'm telling, not the 3 batches, based on the nature of the vendor, nature of the material, you have to evaluate whether the 3 batches or all the batches, and based on that you have to evaluate. Okay. Miladur Rahman is asking, what are the control points or method of risk mitigation if a finished product manufacturer doesn't have the testing resource for NDMA testing? If you don't have the testing resource, then you have to get it tested by the third contact testing laboratory. There are some contact testing laboratory which are the FDA approved laboratories. From there also you can perform the testing. Okay, if you don't have the facility. Please provide the FDA reference where evaluation of excipients is mandatory for finished product manufacturer, and reference of FDA where discussion about NDMA is present. My dear friend, whatever I discuss, it is based on the FDA guideline only. You can search the FDA guideline. There is a very clear message by FDA. Okay. Satish Kumar is asking, packing material sources are not discussed. Yeah, my dear friend, because FDA during this discussion not considered as a packing material, to be very frank, because when you select the packing material, the expectation of a packing material is it should not be reactive, absorptive, additive in your drugs and drug product. So that is why it is not discussed. But definitely due to the packaging material, if there is a possibility of a increase or generation of impurity, then definitely you need to consider this packing material also, no problem. Mr. Pramod Reddy presentation. Yes, I have shared the link, and please go through this link and download the training presentation immediately. Otherwise, this link will expire. And after this training, if anyone is interested in getting training presentation copy, I will not able to issue the training presentation. Okay. So please download it now only. Ria is asking, can you please share some of the excipients that could possess the risk of nitrosamine impurity? Yeah, sure. Even the some examples are also given, that different type of excipients where the amines are used in the excipients and some reactions are there. So that type of excipients, or definitely you can consider the synthetic origins, the API excipients which are from the synthetic origins. There you need to put more and more focus. And second, the other APIs where they are not synthetic origin but they are using some solvents or reagents which are related to the amines, then that also you can consider. Okay. Yes, very good, Mr. Satish Kumar. Nitrocellulose materials also can lead to form impurities. Thank you, Satish Kumar. Thank you for clarifying this response. Okay, I think now 20 minutes, we are ahead to 20 minutes, and there are no any questions. Everyone is clear with the topic. Everyone is received the training presentation. Again, requesting if anyone is a part of a CGMP or advanced GMP member under YouTube membership program, please contact me personally and I will arrange the training certificate for you. Okay, thank you so much, and ensure all time compliance. Thank you so much, and looking forward to see you in our next training session. Till time, thank you so much, and ensure all time compliance. Bye bye, all of you. Bye bye. Now I am closing this session, right? After this, if you have any questions, you can put in the comment section also. After that also, I will respond to you. Okay, thank you so much.